So I'm going to get things started. Our first speaker this morning is going to be Robert Berman, MD. He is the Chief Medical Officer and co-founder of Biohaven Pharmaceuticals, which is in New Haven, Connecticut. Biohaven is a clinical stage biopharmaceutical company with a portfolio of innovative late-stage product candidates targeting neurological diseases. And Dr. Berman has 30 years of experience in neuroscience research, both within academia and industry. Put your hands together and let's welcome Dr. Robert Berman. You're welcome, welcome.
No, I understand. Thank you.
No.
Oh, okay. Better now? Okay, good. It's great to be here. Today I'm going to talk about the clinical trial that Biohaven just completed with our drug Trigriluzole. And you see the complicated sounding title of the talk. And by the end of the talk, I'll parse out all the words in that and you'll understand what a trial is, what that trial is, and also get a sense of what goes in to making a clinical trial happen. We're proud of this effort. It's the first trial that really could serve as a registrational trial. That is, a trial of the quality and size that could be submitted to the FDA that you need in order to get a drug approved. So the first of such trial in Ataxia. A spoiler alert, the trial results don't support immediately going to a filing. But there's some interesting trends I'll show you that show why Biohaven is still interested in this drug and still continuing the study with it. So actually, with the show of hands, how many people participated, were contacted, inquired about it, were screened for this study? I see some hands there, and they're great. Well, I want to thank you all. I'll start by thanking the whole community, because one important thing that this study showed was that we can do trials in ataxia that are required. It hasn't been shown before. And two, that the community is just amazingly receptive to participating in research. And research is what we need to advance the field and get to a treatment and a cure eventually. So we were very pleased with how receptive you all were. So let's dive into it. Complicated slides. So these are the slides actually I'm going to show next door to the scientists in a little bit. But I wanted to, for the sake of transparency, let you see everything that they're going to see. And I'll not speak to all the details of it. But this is about the drug Trigriluzole. Okay, what Trigriluzole gets metabolized, broken down in the body to another drug called riluzole. And this is how it works on the neurons. It works briefly by increasing the neurotransmitter glutamate's uptake and also on ion channels. But in any case, it had an interesting pharmacology that intrigued Biohaven, and I'll talk more about that in a second. The pharmacology being from the drug riluzole, which is actually an old drug, the metabolite. The metabolite's effective in a disease called amyotrophic lateral sclerosis, or Lou Gehrig's disease. But our interest was not for that, obviously, but it had interesting pharmacology. But there are limitations to riluzole that are listed there on the right. There's a food effect, there are effects on the liver, and those were limitations that were addressed by the development of a prodrug, which is Trigriluzole. What a prodrug is, it means that once it gets in the body, our drug, it gets cleaved or metabolized into riluzole and a harmless byproduct. But it avoids the liver, and it doesn't have a food effect. You could take it without regard to meals, and it has a longer half-life and some other properties that make it more amenable to testing in a broader population. So we were excited about Trigriluzole. We're excited about the mechanism, and I'll skip over that. That enthusiasm for the mechanism was shared by a group of Italian researchers. And they actually did 2 studies on the metabolite of trigriluzole, done in 2010 and 2015. And what it showed is that on the left, I won't belabor all the details. On the left, it showed in a diverse group of patients with hereditary and non-hereditary ataxias, you saw improvement within 8 weeks on the scale that's called the ICARS. That was followed on by a study in hereditary ataxia, patients with Friedreich and assorted SCAs on the right. And what you also see is that over 3 months, on the furthest right panel, the blue bar showing the treated patients and the gray, the placebo patients, you see that there's differentiation at 3 months, and that persists and even grows through a year. So that was quite compelling to us, that there are now 2 well-controlled trials with the metabolite of our drug. And so that's when we decided to test our drug for ataxia. So when we set out to do a study, the first order of business is we'll come up with our objectives. And when we think of our objectives, it's really, who's the audience? Okay, well, obviously we want to serve the community of patients with ataxia, right? The other audience is we want it to be convincing to those who would prescribe medications. And then what's less apparent, but very, very important, is the FDA, the Food and Drug Administration, is a key audience, because they're the ones who will say at the end, oh yeah, we approve this drug, you can now offer it to all the patients in the U.S. So those all those considerations get factored into the design of the study. The key outcome variable here was on the scale for the assessment and rating of ataxia, a scale that you may be very well familiar with. But first let me talk about the design of the study. A study is, you hear a lot of science, in here, and I've been here before where the scientists have discussed genetics and in very detailed manners. A clinical trial is more like a wheel, okay? It's simple and very inherently understandable that we identify patients, the qualifying patients, I'll talk about that in a second, they get randomized to either the active drug, which is trigriluzole, 140 milligrams, or placebo, which looks like identical capsules. There's no way to tell them apart. And your investigator doesn't know, you won't know, and even I don't know when the study's ongoing, because even the sponsor, Biohaven, is blinded until the very end of the study. And that is required so that we don't introduce biases. So for our study, we treated patients for 8 weeks in that blinded, that's called a double-blinded, meaning no one knows what you're on, randomized because you're just randomly allocated to a group, and then placebo-controlled because there's the placebo group. So those are the words that were in the title of the study. So they're randomized to that for 8 weeks, and then the patients who complete that 8-week period then get to go into the essentially 1-year extension phase, where everyone at that point is on known drug. And that gives us more information on safety and even on potentially efficacy. So you see there are the outcome variables. There were a lot of different measures. The key one is the SARA. I'll talk, before I focus on the SARA, the patients in this study had SCA 1, 2, 3, 6, 7, 8. And that we limited it to that because the 2 Italian studies that I mentioned before, that's what they focused on. We limited the number, except they didn't have patients with SCA3, and so we limited the number of patients with SCA3. Apologies to those who wanted to get in who had that ataxia. But we limited it because the Italian study didn't have any such patients. And we wanted to stay as close as we could to that study because, in a sense, we're trying to replicate those findings. And then the other principle in a study is that you want the patients who come into the study to be comparable in some ways. So in this regard, the patient's SARA was between 8 and 30, so more the mild to moderate phase. And then there are a bunch of other exclusions. If some symptoms that would make it hard to rate the SARA would cause someone to be excluded. Medical conditions that may confuse how well someone's ataxia is doing, because some medical conditions sort of wax and wane, that would exclude someone. So were some of the criteria that narrow who gets into a study. So let me talk about the SARA. I'm sure many of you are very familiar with this scale. It's an 8-item scale, and your clinician may ask you to walk down a hallway, stand up straight, and they watch your posture and see your sway, or watch you move your finger from your nose to their finger, or chase their finger around, things like that, some of which kind of don't matter a lot to your day-to-day, like maybe the finger chase or the heel to shin. If you were familiar with that, just have your heel follow your shin bone down. But they really reflect a variety of functions of the cerebellum, and that's the part of the brain that contributes to ataxia. So, and that's an instrument that we chose to use in this study to be the primary one, to say, hey, look, our drug worked on this scale and better than placebo did, and that's a win. So let's talk about the study. Get to the end already. Okay, so this is a diagram that shows how many people were screened, how many were randomized, how many completed, and a couple take-home points here. So we had 18 sites in the United States who were doing the study. And the most impressive thing, to me, I'm impressed by you, is that we were able to enroll 140 patients in 6 months. And remember, when a study starts up, it starts slow because not all the sites are up and running. But, and that, I think, will encourage other sponsors as well to come into this area. So that speaks also to the National Ataxia Foundation, getting the word out, the investigators, who are a highly committed group, many of whom hadn't done trials that would serve the FDA before, so it required a lot of learning, but they were game and they jumped into that. So that's the first major success of this study, that we proved that patients will come out and participate. You proved it. So this is some of the demographics, the background information of the patients who participated in the study. And I can't even, I'll face this way for a moment. So generally very well balanced between groups. So that's the way the scientists will look at this is, the placebo group, are they similar to the active group? And they pretty much are. And you see the different SCA genotypes that are included in the study there. And the baseline SARA averaged about 14, which is mild to moderate. Okay, so this is the outcome. What you see in the blue bar is the drug. So on average, patients improved by about 1 point on the SARA. And unfortunately, and that improvement was statistically different than baseline. Unfortunately, placebo also had a similar improvement. So we did not differentiate from placebo on our primary endpoint. So that was a bit of a surprise to us that placebo had such a benefit, because when you think of a progressive neurologic genetic disease, you don't think of placebo response. I'll talk about our thoughts on that in a moment. But this speaks to why we were surprised. So on the far right is our study results. And you see we have the 1 point benefit. The gray bar is placebo's, even a smidgen more, but about 1 point benefit. And on the far left is the Italian study, one of the Italian studies, where they showed a 1 point benefit after 3 months. But the placebo group actually had worsening at 3 months. So that makes it a little hard to interpret the data fully. And then also in there is the responder rate. And the responder rate is between baseline and week 8. How did the SARA, how many patients had a 1-point improvement in the SARA or more? And you see that it was a similar rate in the blue bar in the right panel compared to the gray bar. Again, and that's very different from the furthest right bars, where the placebo group didn't have such a high rate of responders. Okay, so a lot went into this. So we don't stop there. We interrogate the data and try to understand what's going on, and what we learn actually will serve future studies as well. So let me talk about the diagram there. So that tells you how many patients had a change between screening when you first went into the site and baseline, which is the day you got your medications. And that's between 1 and 6 weeks. And you see that about a third of the patients had a 2-point change between screening and baseline. And remember, we're trying to understand if patients have a 1-point or better change. So what that means is just there's a lot of variance, more variance than we were anticipating, even between screening and baseline. So with that, we ask ourselves questions about the data. So the high variability of the SARA scores, that could be due to a lot of things. So we try to understand that. So are there, is there a population, the part who is stable, the 2/3 who are stable between screening and baseline, do they perform differently? Are there some items in the SARA that are more prone to variability? And if we just look at the better items, what does that look like? And then, we only used 1 dose. So we also were able to get a sense of the exposure level, how much drug is in your blood, by looking at some correlates of that. So patients who are lighter or female patients tend to have higher blood levels of the drug. So that was another question we asked. Okay, so let's walk through it. So when we break down the study results by the first 4 items, which are gait, walking, standing, sitting, and speech, versus the appendicular items where you use your fingers and your heel to shin, we see that there is a difference. We see that the placebo effect is really driven by those appendicular items. And actually, if you just focus on the first 4 items, the axial items, there seems to, the trend seems to shift with the drug. And then we looked at the patients who were stable between screening and baseline, who didn't have a lot of variability. And then what you see there is, and I don't think I have a pointer, but the top red square, you see that it favors drug by about 0.4 points. And then if you actually look at the far right there, the number of patients who had a 3-point improvement on the SARA, that's a lot, actually. Then you start seeing the difference really favoring drug a fair amount. So that's interesting to us. We're trying to get around the noise, and we're seeing some signals come out. And then we looked at weight. And so because the patients who are in the lighter half would have a higher concentration. And again, I'll focus you on with the top right, you see that there's a 0.5-point improvement favoring drug. And on the bottom right, you see actually the patients who are lighter and who were also stable between screening and baseline, they also performed very well. So again, that's encouraging in the right direction. And then gender is the same story, and that's because it turns out that females have a higher drug level. I won't focus on that. So efficacy, efficacy means, does the drug work at relieving symptoms? That's one part of a study. The other part is assessing the safety and tolerability. So we did show that the drug was very well tolerated on whole, with few dropouts, and no what you call serious adverse events. So it was the kind of drug that could be tolerated in broad populations. So that was good. So let me summarize what we learned from the study. So one, we demonstrated that these studies can be done, and that other sponsors with other interesting drugs shouldn't shy away from this space because it's a very welcoming community. We showed that the primary outcome variable has more variability than we had realized, and that's important for the next study that we might conduct, or even other sponsors, because there are ways to try to get around that. We showed that there could be a response, an exposure-response relationship that would point us to wanting to look at higher doses in the next study. And so we do have emerging signals. We have the long-term study that's still reading out. The last patient just finished their 6-month visit. So we'll be analyzing that, and then we'll analyze the 1-year visit. And we'll take the entirety of the data, and with that, come to a decision on what's the best course for this drug and the ataxia community. So we're still analyzing this data. There's still other parts that are coming in, actually. And in addition, we're also speaking with the FDA about the data and about the outcome measures, because they're new to this field as well, and they need to understand how we measure severity. So in conclusion, as I started, I want to thank the community here, because the community is hungry for treatments, and they really stood up and volunteered in mass. And then I also want to thank the investigative sites, many of whom you know who've spoken here, because of the tremendous effort that they put into this study, and all the staff at their sites. So with that, I'll turn the podium over, and thank you for your attention. Yeah. Thank you.